What if I told you that two human parents are capable of producing more than 65 trillion genetically unique offspring? That's a lot of babies, and that's a lot of sleepless nights, and it begs the question, how did just two people generate so much genetic diversity? Well, that's what we're going to explore. First, we need to take a step back and consider that in the human body there are a whole range of different types of cell. In fact, there are about 200 different cell types. The thing is, we can classify any cell within the human body into one of two types. It's either what we call a somatic cell, or it is a gamete. Now, nearly all the cells in your body are types of somatic cell. The exceptions here are the sperm and the egg. So if we're talking neurons, or your muscle cells, or skin, or bone, or liver, or kidney cells, these are all types of somatic cell. And when a somatic cell wants to divide, wants to reproduce itself as it may need to for either the purposes of growth or tissue repair or regeneration, it does so by the cell division process that we typically just refer to as mitosis. Now, if we look within the nuclei of our somatic cells, what we'll see is there are 46 chromosomes. And before mitosis can occur, we need to replicate all of those chromosomes. And when that process is finished, each chromosome then exists as two identical copies held tightly together that we call sister chromatids. Then during mitosis, well, those sister chromatids are separated, the cell will physically divide, and those resulting daughter cells then have their own set of 46 chromosomes, and importantly, they are genetically identical. But if we look a little closer at those 46 chromosomes, what we'll actually notice is that they're composed of pairs, 23 pairs to be exact, and we refer to them as homologous pairs. Now, the word homology in a simple sense just means similarity. And we call these homologous pairs of chromosomes, well, because both chromosomes in a pair carry the same genetic information. In fact, they have exactly the same genes in exactly the same locations. So an important question then is, how do we end up with pairs of chromosomes in our somatic cells? The answer is that you inherited one set from your mother and one set from your father. In other words, you have matching sets. This arrangement of chromosomes is referred to as diploid. Diploid is a very important piece of terminology, but what it actually really just means is that you have two sets of chromosomes. And the ultimate reason for this is that we are the product of sexual reproduction, meaning two parents giving rise to a unique combination of genetic material through the process of fertilization, or the coming together of sperm and egg. The two types of cell that we do not describe as somatic, but instead we refer to as gametes, or sometimes just simply sex cells. Now, When the sperm and egg come together, during fertilization, what they produce is a zygote, a single somatic diploid cell that will undergo countless rounds of cell division by mitosis, an extensive cellular differentiation to give rise to you. Now, let's just stop and think about this for a second. That zygote is diploid. So it has 46 chromosomes, or 23 pairs, or 2 sets. Well, that must mean, therefore, that the gametes, the sperm and the egg that fuse to produce that zygote, must have only had 23 chromosomes, or in other words, must have only had a single set. And indeed, that is the case. And for this reason, we describe gametes as being haploid, meaning that they contain only one set of chromosomes as compared to the somatic cells, which contain 2. And in fact, this is a key feature of all life cycles brought about by sexual reproduction. A haploid sperm meets a haploid egg and results in the production of a diploid zygote. And this whole cycle is able to continue when you grow and reach sexual maturity, and you are then able to produce your own haploid gametes. Now, remember that when somatic cells divide, they produce genetically identical diploid daughter cells. So this must mean that there is some alternative mechanism of cell division, some alternative mechanism to produce haploid gametes. That mechanism, that cell division process, is called meiosis. Now, as we're going to see, meiosis does actually begin with a diploid cell, but just think about what would happen if we started with a diploid cell and then produced gametes using mitosis. Well, what that would mean is that the gametes would be diploid. They would have 2 sets of chromosome, and when the sperm and the egg would come together, you would get a zygote with 4 sets. Well, that's actually something called tetraploidy, but we know that's not the case. Instead, we have this mechanism that starts with a diploid cell and results or ends up with haploid gametes. Then a haploid sperm meets a haploid egg and results in the production of a diploid zygote. In addition to reducing the number of chromosome sets from diploid to haploid, meiosis also contains a series of mechanisms that ensure increased genetic diversity. So, as we work our way through meiosis, as always, it's not the details that are so important, it's some of the bigger picture questions. Things like, how do we go from a diploid starting cell to a haploid gamete? Related to that, why does this require 2 divisions to achieve this? And then, as I just mentioned, where within this process is genetic diversity generated? Now, as we're going to see, meiosis starts with a diploid cell. And in the testes, when producing sperm, that cell is referred to as a primary spermatocyte. In the ovaries, when producing eggs, it's referred to as a primary oocyte. Now, for illustrative purposes, we're just going to show you a cell with 4 chromosomes or 2 homologous pairs. But rest assured, the same thing is happening to the rest of the chromosomes within the cell. Now, just like mitosis, meiosis begins with replication of the DNA. And again, by the end of this process, each one of those 46 chromosomes will now consist of 2 identical sister chromatids. What we're going to see happen in the process of meiosis is 2 divisions are going to take place, meiosis 1, meiosis 2. Those 2 divisions of the first one, the homologous pairs of chromosomes are first going to be brought together and then separated. In the second division, the chromatids will be separated, giving us haploid gametes. And as I just said though, there's more to meiosis than simply reducing the chromosome number. There are 2 major mechanisms for generating diversity. One of these is called recombination, and it's where homologous chromosomes will actually exchange corresponding sections. The other is called independent orientation. Amazingly, this just refers to the way those homologous pairs line up at metaphase. Okay, so let's make our way through. Meiosis 1 begins with prophase 1. During this phase, the chromosomes will coil, they will condense, and they will become compacted down to their most compact form. What follows is a super cool event. The homologous pairs of chromosomes are then brought together. This process is called synapsis, the details of which really aren't important right now, but I just want you to understand that you are bringing those pairs of chromosomes into alignment with each other. Now remember that each one of these chromosomes consists of 2 chromatids, so because that means we have 4 chromatids in total, we refer to this as a tetrad. And when you align these tetrads in this fashion, they will actually swap corresponding segments of DNA. This is called crossing over. Now, these are homologous chromosomes, remember. You inherited one from each of your parents. So they have the same genes in the same locations. But for every single one of those genes, within our population, there are different versions. We call these versions alleles. They differ from each other just by small changes in DNA sequence. In fact, sometimes it's only a single nucleotide change. What this means, though, is that as a result of crossing over, what we've generated are composite chromosomes. Chromosomes that are a composite of maternally and paternally inherited DNA. And what this really means is what we've created here are unique combinations of alleles, combinations of your father's and your mother's alleles. And in fact, if you look at those 4 chromatids in the tetrad, you'll notice that each one now is genetically distinct from the other. So that's a crossing over event. Actually, as it turns out, crossing over occurs between 1 and 3 times randomly along the length of each set of homologous pairs of chromosomes. This crossing over is therefore a major source of genetic diversity. Now, as meiosis progresses, what we're actually going to see happening is that the tetrads are going to move into the middle of the cell to the metaphase plate, and those homologous pairs are then going to be separated during anaphase. But we have to hold on for a moment here, because the way that those tetrads align themselves in the middle of the cell is actually our second major source of genetic diversity. This is actually what we're talking about when we say independent orientation. You see, each homologous pair of chromosomes orients itself or aligns itself independently at the metaphase plate, meaning there's an equal probability of either the paternal or maternal chromosome facing either pole of the cell, again, which means then it's totally random which chromosome goes to which pole. Now, let me try and illustrate this with, again, just 4 chromosomes, 2 homologous pairs. They have been brought together. They have exchanged corresponding sections during crossing over, and they have now aligned at the metaphase plate. Well, let's take a look at our top tetrad. It can face either direction, or if you like, it can face either pole of the cell. And the thing is, the same is true of the bottom tetrad. It could be facing either pole of the cell. The point is, the alignment of one tetrad, or to put it more accurately, the orientation, is completely independent of all the other tetrads. So what that means is, in our simple example here with just 2 homologous pairs, if we run the process of meiosis forward a bit, what we're actually able to see is that with just 2 pairs, based on nothing more than the way that they align in the middle of the cell, you can generate 4 different combinations. But hold that thought, because we're going to come back to that in a moment. Now, let's just focus on one of these combinations and see how meiosis proceeds from this point. So the homologous pairs will separate during anaphase, and the chromosomes will move towards the poles of the cell. Cytokinesis will follow, which will split the cells in half. And what do we now have? Well, now we have 2 haploid cells. Remember, we brought the chromosome pairs together, they exchanged corresponding segments, and now we have separated them into separate haploid cells. The thing is, though, those chromosomes, remember, consist of 2 chromatids. And so this is why we now need a second division. So as we move through meiosis 2, really the key thing to appreciate is that these chromosomes now align on this metaphase plate. The chromatids are now separated. Nuclei will form around the DNA. Cytokinesis will split. And what do we now have? Now we have haploid gametes. So that's the process of meiosis, a very, very cool series of cellular events in which we saw a diploid cell go through 2 divisions to give rise to haploid gametes. And along the way, we saw how diversity is generated by homologous chromosomes exchanging segments, generating new allele combinations, and the independent orientation of those tetrads, of those homologous chromosomes, how that in fact generates diversity all on its own. But 65 trillion? That's quite a number. But come back to independent orientation. I showed you with our cell that with just 2 pairs of chromosomes, we can generate 4 different combinations based on just nothing more than the direction they face when they line up at the metaphase plate. But as humans, we don't have 2 homologous pairs. We have 23 homologous pairs. And so, if you want to know how many combinations are possible, you have to do 2 to the power of 23. Turns out, that's 8.4 million. So in other words, every sperm is carrying one of a possible 8.4 million combinations of maternal and paternally inherited chromosomes based on independent orientation. Well, if that's happening with the sperm, it must be happening with the egg as well. And indeed, the egg is carrying one of a possible 8.4 million combinations of maternal and paternally inherited chromosomes based on nothing more than independent orientation. But here's the thing, fertilization is random. It is random which sperm meets which egg. So if you want to know the total number of possible combinations, you have to take 8.4 million and times it by 8.4 million. You know what that gives you? About 65 trillion different combinations. And that doesn't even take into account the fact we've generated new allele combinations through the process of crossing over, or that random mutations will have occurred during the replication of the DNA. So the next time somebody says, "You're awesome, kid, you're 1 in a million," you're far more special than that. You tell them, "I'm 1 in 65 trillion.